Hi, I'm Jacob Welch. I'm in Chem 231, and this is the identification of unknowns using infrared and HNMR spectroscopy. The objective of this lab was to understand the principles underlying infrared spectroscopy and proton nuclear magnetic resonance spectroscopy. To obtain IR spectrum using the salt plate method as well as the HNMR spectrum. To analyze and interpret the spectra to determine the identity of each unknown compound. In an announcement from the Ohio Department of Health, citizens of Alliance were asked to bring all unused or expired medications to the Alliance Police Department for collection and proper disposal. This announcement stems from research demonstrating that the active ingredients of medications can be found throughout the environment when improperly disposed of. In this experiment, we helped the Ohio Department of Health determine the identity of some unlabeled medications. The methods used for obtaining an IR spectra were to obtain an unknown solution and place a droplet of the unknown on a salt plate, then sandwich it together with another salt plate. This was then placed in the IR reader. The results were then computed and graphed. These were the results of the IR spectroscopy. The test yielded two significant peaks, one at 2981.13, indicating a CH bond sp3 hybridized. There was another peak at 1715 indicating a CO double bond. This could be an aldehyde, ketone, or carboxylic acid. These are the possible molecules of group 6. According to the peak here, there must be carbon to hydrogen bond sp3 hybridized. All three of the molecules exhibit that type of bond. According to the peak here, there must be a CO double bond. This eliminates this molecule and this molecule because they do not exhibit this bond. That means that T-butanone must be our unknown substance. The methods for proton NMR are as follows. First, you obtain an unknown solution, then add roughly 0.5 milliliters of CdLCl4. Then you add 0.5 milliliters of the unknown solution. These two substances are then mixed together and placed in an NMR tube. This tube was then placed in an NMR reader. The results were computed and printed. The first peak is located at 5.62 ppm. The splitting pattern is difficult to ascertain. The chemical shift indicates a proton bonded to an alkene. This peak has an integration of 1. The next peak is at 1.9 ppm. This chemical shift indicates an allylic proton. The splitting pattern is also difficult to determine. This has an integration of 2. The next peak is at 1.579 ppm. This chemical shift indicates a proton attached to a saturated carbon far from other functional groups. The splitting pattern on this is also difficult to determine. This has an integration of 2. These are the possible identifications for the unknown substance. Looking at the NMR spectroscopy, there is no evidence to suggest there is an aromatic compound present. This eliminates this molecule, this molecule, this molecule, and this molecule. There is also no evidence to suggest the substance is a carboxylic acid. This eliminates this molecule. Since the first peak is located at 5.62 ppm, this indicates a proton that is bonded to an alkene. This means there must be a C double bonded to a C within our molecule. This eliminates this molecule, this molecule, this molecule, this molecule, this molecule, and this molecule, leaving the only possible identification as cyclohexene. The first chemical shift at 5.62 ppm corresponds to these two hydrogens. They are both bonded to an alkene, and there are 2 of them. The next peak at 1.9 ppm belongs to an allylic proton. These are protons next to alkene groups. This corresponds to the 2 protons on each of these sides, with a total number of 4. The next peak at 1 ppm corresponds to hydrogens attached to a saturated carbon far from their functional groups. This would correspond to these two sets of hydrogens being 4. When this ratio is reduced, it comes out as 1 to 2 to 2, which matches our integrations on the NMR spectroscopy. This unknown substance must be cyclohexene. This is my reference. Thank you for listening.